Perfect. All right. Thanks, Laura. Let's see. I just want to say hi. We got about 33 so far and 70 registered. So that's exciting, just having that much interest. But I just want to say hello. I'm Marnie Warren. I'm the co-president with Brian of CCC, and I want to thank you all for being here today. It's exciting to see so many in our community that are engaged and really want to learn. So our goal really is to get you guys closer to the science and just make you aware of all the research that's being done and all the opportunities that will be to participate and ultimately just empower our community. So, but before we dive in to the research, Brian, if you want to bring up the agenda, I just want to, that's what we're going to talk about today. I'm going to do a welcome and then I'm going to turn it over to Brian to dive into the research after I'm done with my welcome. And he's going to take us through a path of funding research towards a cure. But before we get in, I want to take a step back and just quickly reflect on the growth of our organization. I don't know if you guys have seen this in our newsletter, but just in the last year, we've doubled. We're at 333 registered patients on our website. So that's exciting. We almost every day have a new registrant, but it's growing fast, and that just tells me and tells us that there are a lot of people out there that that haven't been diagnosed yet, and then that our community is going to continue to grow. And it's just that, and it's just going to be important that we all stay together as a community and we stay organized and we together push for more research. And what we've learned is that while research is critical, but so is the support of the community. It can be really lonely, as you guys know, when you first get that CHD2 diagnosis. Our goal is to alleviate some of that fear and support you with the information and help everyone overcome that initial diagnosis. So, but there's only 8 of us, and we're all of us on the board. We are either a parent or a sibling of someone with CHD2 or a rare epilepsy, and so this disease is, it impacts all of us, and we all have full plates. So it's going to take all of us, our entire community, to really push this forward. So because of that, Brian, if you will go to the next slide. While we were all together at the research, after all the researchers left, we took the opportunity to put together our first strategic and research plan, which are finalized finally, and they'll be up on the website soon. So I really want to encourage you to go look it up and look at and go through it and look for opportunities to get involved. We've already had at least 10, I didn't count exactly, but 10 new committee members that have joined and become participated in our committees. And it's just been fabulous to see everyone jumping in and helping out, because we have a lot to do to be successful and reach all our strategic goals. So with that, we have a new mission statement that was originally focused on the research, and now it's become, we want to include more community. And so we have our 3 pillars of community and research as our priorities. But putting this together, I don't know if you remember, we did a community needs assessment, which we used to help guide us in this for this strategic plan. And from that, I think we had 132 respondents. And the number one thing you guys told us was that you want us to focus on funding scientific. So that's what we're here today, is to show you that what we're doing so far. So and I wish I could say that now that CHD2 has been discovered that it's going to be easy, but unfortunately CHD2 is very complicated. It's essential to the development of the brain, and thankfully there's a lot of traction going on in CHD2 research, but not only because it just helps us understand our underlying biology, but also because CCC is, we've been the cheerleaders out there, letting the researchers know that we have a community, we have a patient advocacy group that's devoted to understanding this gene, and we're the ones, we have a support of an entire community that's going to jump in there and participate and help out, because it's going to really take all of us to get the research forward. So I encourage you to go look at the research plan and the strategic plan online. It should be up soon. And the researchers, they were excited. They're taking the time to pull together and find ways that we pulled them together and find ways to collaborate and share their research. It was really exciting to see all the various research that you're going to hear about today. And they left enthusiastic, and we've now put together a plan for them, and they've helped put that plan together. And so I want to say, I'm going to do a quick plug too that, we hope to in June, we're almost there, signing on the dotted line in June 2025 to have our next research and family conference. So I want to really encourage all of you to come and join and meet all the researchers, because all of these and more will probably be there and invited back to share what's the continuation of the research. So be looking for those details, and I really hope you guys can come. So that would be the last one we had at Disney was awesome. So I'm really to announce the next one. But before we move on, I wanted to do a quick video for you. I just wanted you to kind of get a feel of being there with the scientists. I want to preface it by saying that, when we were there, we were outside in the evening, and out of nowhere, a SpaceX rocket launched over. Gianna, you're probably used to this, but you people in Florida are used to this. But those of us not from Florida, suddenly a rocket appeared, and it was really exciting. It kind of got everyone kind of excited it was like the first night. And so therefore the theme of the weekend became Rocket Man. So if you want to go ahead and hit that really quickly, and I just wanted to give you a quick synapse of our weekend.
I engage with CCC because I can see that they've got a big fight and they need the help they can get. I'd like to think I've got something I can contribute there. It's an epilepsy that is difficult to treat. It's not just seizures. There are all these other symptoms that affect people and have secondary effects on their caregivers, their families. To me as a doctor, I want to go to bed at night knowing I've been trying to fight the good fights. And I think this is one that really does merit making as much effort I can to help people. And as I've been chatting with my colleagues here, when I retire one day, I hope that I can look back and say, we contributed our little bits to help in this fight and hopefully left things better than we found them.
Research takes time. Unfortunately, people say, and I think it's true in my own experience, that 90% of the work that is done in some sort of experimental context fails. And the only thing you'll hear about as a funder or as someone who follows science in the news is the results of experiments that worked. And in between that time, you have no idea how many experiments failed.
I think it is very important for parents and caregivers of adults to be involved in the research and be involved with the clinical side, knowing what is happening all the time.
A huge thank you to all the families who participate in those kinds of materials, because now there's this personal connection and there's this drive to really execute fantastic research. And I mean, before you could still execute fantastic research, but when you know the mission behind the initiative, behind the DNA, it's super, super impactful for us as researchers.
Great, thank you, Laura, for running that. Sorry, that was a little technically glitched, but I think you guys got the hint that it was a really exciting meeting. The researchers were really excited when they left. They can't, we really look forward to seeing them again, and we've continued conversations with them, and you can see that, and Brian's going to go over that. But I really quickly wanted just to say, I know there will probably be questions along the way. You're welcome to put them in the chat, and Laura and I will try to get to them either during the session or at the end. We hope to have some time for some questions at the end. So But I really, again, want to thank you guys all for being here, and and I look forward to sharing everything with you. So Brian, I'm going to turn it over to you.
Thanks, Marnie. I'm Brian Broadbent, the co-president along with Marnie. I live in Dallas, Texas. It's really a pleasure to be in front of you all today. I'm here to kind of try to break down what happened at the meeting of the minds. Most of the people on this call are not scientists. And so I think before I hop into what happened, it's just important to go through a couple of principles when we think about a cure that are really important. And also, since we're not all scientists and I'm not a scientist, I try to break down science into kind of simple real world analogies that help me kind of understand things. And some of them are corny, and I apologize in advance. So the first thing for any essential treatment is that there has to be a measurable clinical benefit. There has to be a clinical endpoint. There has to be something, a measurable improvement. And so it's not enough. If you look at this ad for Pantene, it's not enough to say, wow, this made my hair feel better. That's not good enough. Here's another ad. It says, train your hair stronger. That's better. There's a benefit, but there's no data. And if you look on the right, it says thicker, stronger hair in 14 days. That's what the FDA is looking for. They want something that's measurable and quantifiable. And so for CHD2, the most common thing might be seizures, but there could be all kinds of clinical endpoints that measure improvement from a treatment. There could be behavioral measures, communication measures, and there's all sorts of things, and there's really no magic rule of what it is. It just has to be something that's measurable. There's kind of two main phases and I'm using the FDA here in the United States because it's just the easiest example. There's two main phases towards kind of getting a treatment in your kids. The first is there's what's called an IND application, and that stands for investigational new drug. And in this phase, you're really proving a benefit in animal models and animals, although cells are starting to come into that, and we'll go into that later. And then after you get indication for to start this trial, then you look to do clinical trials in humans, and here you're looking for human data. And so the common theme here is you're looking to collect data, and establishing rigorous data is expensive, but that's really what the FDA is looking for. So another thing that I think is important is that, there's some challenges with CHD2. And one of those is that CHD2 is a chromatin remodeler that impacts how other genes function. CHD2 disruption results in a lot of little changes and therefore a variable phenotype. What that might not mean much to a lot of people on this call, but to try to sort of simplify it, think of a chromatin remodeler like CHD2 versus some other traditional genes you've heard of before. And sort of that analogy I like to use here is a factory floor. And many genes in our body, they play like one key function. And so on the assembly line, you could see a traditional gene might be responsible for placing the glass in that car. CHD2 is more of a chromatin remodeler. It doesn't play one little piece, but it sort of plays a piece in the entire assembly line. And so there are many different chromatin remodelers. And so I sort of, in this example, I'm saying CHD2 is responsible for the lubrication of all the machines you see, and some you don't see in this factory line. And so if CHD2 doesn't work, then these machines do not get lubricated and they're going to eventually break down. And you sort of don't know which machine's going to break down first, because each factory is different, and all our kids are kind of, our bodies are their own little factory lines, but the machines will eventually break down. And so that just kind of leads to variability. The second problem is that CHD2 is what we call a Goldilocks gene. And so too much is also harmful. And my daughter is an example of that. And so when we think about treatments for CHD2, we have to get it to the right level, like not too little or not too much. So there will not be a quiz on this chart. Another challenge is that it's very important in the body for there to be precise levels of CHD2. And in this chart, you'll see a very complicated chart from fission yeast. And this was done in a paper, and it was sort of comparing this fission yeast to CHD2. And in the chart above, you see a lot of things going on. You see all these genes happening. And in CHD2, we think a lot of this regulation of CHD2 is done by chaser. And so, but chaser and CHD2 are they're sort of replacing the function of all these other genes that you see above. And so it's a very intricate process. And so that we don't fully understand. And there actually may be other genes that could play a role in this. And so it's just complicated. Another challenge, you may have heard of gene therapy, and the problem with CHD2 is that it's too large for traditional gene therapy. For gene therapy, they use these things called viral vectors, and they can only hold so much. And so and CHD2 is too big to fit in the current vectors. And so there's a lot of people are working on a lot of ideas and strategies by potentially doing two vectors and putting them together, or mini genes. And there's scientists trying to come up with these solutions. But right now, CHD2 is too large for traditional gene therapy replacement. So we have to think of other strategies. The final challenge, and this really isn't a CHD2 thing, it's really with all neurological disorders, is that CHD2 impacts the brain. And so there's this thing called the blood-brain barrier that we all have, and it sort of protects our brain. It protects things from our blood getting into our brain. And so when we think about treatments, we have to think about how to get around this blood-brain barrier in order to deliver treatments to the brain. And again, this is not there are scientists coming up with all sorts of ways to kind of creatively get stuff to the brain, but it is a challenge. And, despite the challenges, there are a variety of therapeutic approaches to correct CHD2 to optimal levels. And, there's a lot of great people working on these solutions. And so that kind of segues into, okay, what happened at the meeting of the minds? And so there are kind of four key. So there is a lot of stuff that happened at the meeting of the minds, and it's really too much to pack into one presentation. So I tried to break it into kind of four key takeaways. And the first thing that happened was that there was by bringing people together, there was the groundwork for a future natural history study in CHD2. And what are natural history studies? What's a natural history study is a pre-planned observational study intended to track the course of a disease. And so what this really means is, we're trying to develop an understanding of what it means to have CHD2, with the ultimate goal of establishing goals or endpoints for a future therapy. And to create a measurable goal, you need to know where you're starting from. And so it's sort of the simple analogy is like in a typing class. If you've ever taken a typing class, you always, at the beginning of a typing class, you figure out what you can do words per minute at the beginning of the class, and then you measure it at the end of the class. A natural history study is like that times 10,000. And so at the conference, first we had Heather Medford come in, and she talked about the work. She's from St. Jude. I know a lot of you have already spoken with her team. She kind of talked about what she's doing. They're also kind of trying to collect data from the online registries. She presented some data on the pediatric side. I'm not going to really go through this data. We also had Dr. Danielle Andrade from the University of Toronto come in. She's focused on the adult side. The adult side is very important because we all want to know what the future holds for our kids. And so she presented her work on the adult side. And again, I'm not going to really go through this. But the key thing is we brought these people together, and there's been a lot of conversations that have happened after the meeting about a future natural history study for CHD2, and we're real excited about it. And so the key thing with that is there's going to be a time where we're going to need you to volunteer and to participate in these studies, because without your kids and their participation, these studies are meaningless. So that was kind of key takeaway one. The second key takeaway was there was a robust discussion on animal models for CHD2. And so you say, well, what are animal models? With animal models, you create a disease state you aim to study in an animal that also contains the same gene. So you induce a problem with CHD2 in a mouse or in a zebrafish or a frog. And then you measure what happens with the disease versus controls. And why are animal models important? It really helps you understand the disease biology. And to get to the IND phase, we talked about this earlier, you need to establish the treatment first works in an animal model. For CHD2, the ideal model has seizures, and we're going to talk about how that's kind of a problem for us. And so the first thing, Moran Rubenstein presented some data on her CHD2 mouse model, which she characterized through a CHD2 grant along with Igor Litsky at the Weizmann Institute. And she presented data on her mouse. And one of the things that she's seen in the CHD2 mouse is that she sees reduced EEG power. And it's sort of, if you look kind of in the lower right corner, you see some changes, and they're changes, but they're probably not meaningful enough to be used for a clinical trial. And then the other thing with the mouse is they don't have seizures even after we try to induce them. They do, however, have motor deficits, and this is a test here called the clasping score, and there's all sorts of behavioral deficits that they try to elicit in these mice. And so these deficits, they're mild, but they might be enough to be IND enabling, but it's sort of at the lower level of what the FDA might approve. Another challenge with these motor deficits is that with the mice, we tend to see them pretty early on in life. And so over time, the deficits become less significant. And so you really have to if you were going to test a treatment on these mice, you have to test it very early. And so one sort of concern, and this is a bit controversial, is that it's unclear whether our current mice have a true reduction in CHD2 protein. And if you look at the right side, the furthest right column where the arrow is, that's where a mice has it's both of its copies of the CHD2 gene are defective, and you still see some CHD2 protein there. And so that may be evidence that this mouse model, the mutation that they're inducing, which is very early on in the protein, may not be a full reduction. And so we may need to do another mouse model in the future. And so there are things other than mice. And so Chris McGraw, who you saw in the video, he gave a presentation on using zebrafish as a model for rare epilepsies and other neurological disorders. And I'm not going to go into too many of the details, but zebrafish are really cool. And one of the nice things about using an animal like zebrafish is you can make stuff happen faster than zebrafish. You can make things happen faster in zebrafish than you can in mice. And with zebrafish, there's two main approaches towards zebrafish development. You can create a genetic defect in the animal, and I kind of call this like pulling the wire out of a computer, like you're unplugging the computer. You are trying to create a genetic defect in the animal that mimics a genetic mutation. The second thing in these animals with zebrafish, and you can also do it with frogs, is you can do something called a morpholino. And with the morpholino, it's kind of like the picture on the right shows. You just basically kind of use a hammer and you knock down the CHD2 protein. And it's easier, it's often easier to do than the genetic defect. And you can get results really fast. And the potential drawback is they're using this hammering approach. You can kind of create possible secondary effects. And so it's kind of debatable. There's some pros and cons on what would be the best approach, but sort of the key takeaway here is that we have some good results from the CHD2 mouse model, but the community could benefit from a better animal model. Okay, this next section is really cool. The third takeaway was about iPSC organoids for CHD2, and this is what I call brain on a dish. And so you're like, okay, what the heck is what is an iPSC? And so what an iPSC is it's really a cellular model of CHD2. And so what scientists have figured out how to do is they take cells from an impacted patient, and the best is probably skin fibroblasts, but they can also do it with blood and urine and hair and some other cells as well. And what scientists can do is they can take these cells and they can reprogram them back to an embryonic state. And then from that embryonic state, they can grow cells into different types of cells that you see in the body. And really, I have a magic wand there because it's almost kind of magic. and they can grow blood cells, brain cells, pancreatic cells. It's really cool. And so why are cellular models important? Humans are not mice or zebrafish. You know, we're different. We have genetic differences. And so, you know, we really want to understand how CHD2 impacts real human cells. And more recently, there was the FDA passed the FDA Modernization Act 2.0 in December of 2022 that allows these models to be used as substitute for animal models in certain instances. And we sort of expect the permitted uses to grow. And so we had Dr. Nael Khosry from the Netherlands. He talked about the work he's doing on CHD2 in human stem cells. And so, you know, one thing that he pointed out with CHD2, and in most epilepsies, we think about neurons. If you read a lot of research about epilepsy, you're going to hear, and you get into the science, you'll see neurons. But actually CHD2, if you can see in this chart, the red is where CHD2 is most involved, and you can see it's most expressed in actually microglia. And so what he's doing with this work is he's taking CHD2 cells, and he's studying CHD2 in all sorts of different types of cells, not just neurons. You see on the left, you have glutamatergic, GABAergic, I can't even pronounce it. These are all types of neurons. But over on the right are astrocytes, microglia, different types of cells. And then it takes several months, but what you can actually do is you can actually grow these cells into what are called organoids, and they're literally like mini brains. And just studying these can be really helpful to understand CHD2. And so not only can you grow out these cells, but you can use these things called microelectrode arrays. They're called MEAs. And it's almost like you can almost do like an EEG on these cells. And you can look for, it's like they're not seizures, but it's irregular brain activity on these cells. And so the logical question is, you know, what do we see on CHD2 cells? And what he actually sees is he sees aberrant activity on CHD2 cells. And it's sort of hard to see, but basically these waves back and forth are kind of like seizures, and they're different from controls. And then so what we can do is we can then test anti-epileptic drugs against these cells and see, hey, can they bring this wave that you see here back to normal? And so it's really exciting and fascinating stuff. And so he's done these on neurons, but now he wants to extend this work to these organoids as well. And so it's just really kind of fascinating. And I said before, like the FDA is now accepting, at least for drug screens, like this could be a model to bring a treatment to market. And so really exciting stuff. And so in fact, he is testing various strategies in these cells to see if he can bring CHD2 levels to normal levels. So really exciting, fascinating work, more work to be done, but just kind of amazing that science can do things like this. So that's brain organoids. The fourth takeaway from the meeting of the minds was we discussed different possible treatment options for CHD2. And, sort of why are treatments important? It's kind of in the job description as parents to empower our kids to have the best life possible. But as Marnie kind of said earlier, this disease doesn't only impact our child, it impacts other members of the family. And in thinking about treatments, we need to be hopeful but realistic. A treatment might not fully reverse the symptoms, but it can still greatly increase quality of life. So what are some of the possible ways to treat CHD2? The first is what's called an ASO, an antisense oligonucleotide. And what is that? It's a type of molecule that directly targets a specific sequence of messenger RNA to inhibit or enhance function of that RNA. And it's typically delivered via the spinal cord to get past the blood-brain barrier, although there's scientists trying to figure out ways to not have to do that. And you may have seen some of our webinars before with Igor Litsky from the Weizmann Institute. This work was sponsored by Mazi Therapeutics, and he has a philosophy. He has a hypothesis on how to use the lincRNA chaser to increase CHD2 expression. And to kind of try to explain this in simple terms, how to get to the end goal. On the top, you can see this is what they call the wild type. Just you and I, regular people without impacted CHD2, and you have two copies, and one copy produces some, the other copy produces some. And then below that, you have people with which is my daughter, that have an issue with chaser. And you can see their bottom copy is normal, but their top copy produces basically double. And that also causes disease. And then in the third box, you have individuals with CHD2 haploinsufficiency, and one of their copies produces nothing. And the other copy produces a little bit more than 50%, but it's close to 50%. And so what his hypothesis is that he can use an ASO to try to drive down chaser to increase CHD2 expression. And one of the things that he shared that is really important is we don't really, my daughter is an example, loss of chaser also causes problems, and there may be additional problems beyond CHD2. And so just sort of like, if you saw before, we had that hammer and we showed whacking the computer. So what he didn't want to take the approach of just, I want to whack down chaser. He's like, I want to figure out what part of chaser, what's a smaller part of chaser that I could target. And so he, along with a postdoc, they developed this technology, and they looked at chaser across all of these different animal species and found like very short parts that were what they call conserved. Like in evolution, they say what parts of genetics are conserved are the most important. And so he found, and there were actually not that many spots. And so he started to test them. And eventually he came out with one part of the genetic sequence in the 5th exon of chaser that seemed to be really important. And one other thing from his work is they knew that by targeting chaser that they could increase CHD2 expression, but they really weren't sure how it was going to happen. And what they found was that in normal humans, you and I, you have chaser and CHD2, and most of the time you just produce CHD2. About 1% of the time, a little bit of chaser sort of fuses to CHD2 and replaces the first exon and looks like 99% of what normal CHD2 protein is. And basically what the ASO does is it increases this, they call it this chaser CHD2 fusion transcript by about 100-fold. And it's this little piece that you're increasing 100-fold. And when you put it all together, it kind of beautifully increases CHD2 levels by about double, which is exactly where we want it to be. And what's also nice about this is from a therapeutic standpoint, what they call this is alternative splicing. And if you've seen other ASOs come to market, like there's one for spinal muscular atrophy, Spinraza, it involves alternative splicing, and it's sort of like a proven way of an ASO working. And so sort of after the meeting, they've seen some benefits of, it's not really in this chart, but actually improving some of these behavioral measures. And so, it's really, it's really exciting. They've made a lot of great progress. I'm not going to go through all of these benefits, but they're seeing good results from it. And it's really, really exciting. So,
Brian, I'm going to interrupt for 1 sec because there was just a question about what chaser is. And I don't want to say it wrong. So would you just explain that it's the gene
Sure.
next to CHD2?
So what chaser is it's called a long non-coding RNA, and chaser sits just upstream of CHD2, and basically what chaser seems to do is it keeps your body from producing too much CHD2. It sort of regulates it so you're just at the right level. And so what happens if you miss one copy of chaser, you produce too much CHD2, and that causes disease. And so really chaser is a link RNA, and so what it does is it doesn't produce a protein. Its job is to kind of regulate CHD2. Does that make sense?
Thank you.
Okay. So that's one potential therapeutic option for CHD2. Another one is called, you can see the title of the presentation I put on the right. This is CRISPR-A, and this is from Nadad Ahatouf from UCSF. And CRISPR-A is pretty cool as well. And so, sort of already covered this before, but we talked about haploinsufficiency and kind of what that means. And this is like a really good graph that you can see figure. On the top, you have normal state. You have 2 good copies of CHD2, and your gene levels are at full capacity. On the bottom, you have someone with CHD2 haploinsufficiency, that one of their CHD2 copies doesn't work. And so what CRISPR is a lot of times you think about CRISPR, and if you've heard of CRISPR, you think about cutting genes. That's not really how CRISPR is being used here. They take away the cutting. And the other beautiful thing about CRISPR is it allows you to really target specific things in the genome. And so what CRISPR-A does, the A stands for activation. And there's these genetic sequences that exist in the body that they call like activators and enhancers. And what you're trying to do with CRISPR-A is you're trying to deliver this cargo via CRISPR to this part of the gene called a promoter to basically get the gene to produce more. And so what you're trying to do is, and I'm gonna show a sort of a graphical example, a picture to try to illustrate it. But what you're trying to do is get that one copy that works normally to produce more. And so I played this video game back in the 1980s, and sort of to try to explain how this works. In this video game, you can kind of see on the right side, there's two buttons, run, and to make this game work, you had to hit the buttons like and to go faster and faster. And so in CHD2, one of the buttons is broken. And so you with CRISPR-A, you're trying to make that one button that works, you're trying to press it even faster. And the better analogy that I have is that there's these parts in the genome that they call them enhancers. And I think of it as like a coxswain on a boat that says, hey, row faster, row faster. And so with CRISPR-A, you're trying to find something, an enhancer, a coxswain, and deliver them to the boat to get the rowers to row faster. And so with CHD2, basically they've looked all over the genome, and they see a promising enhancer for CHD2 that could possibly make this therapy work. Like the work hasn't been done yet, but it is a promising opportunity. And there was another researcher in the Netherlands that has also found the same enhancer. And so it is another possible way to increase CHD2 levels. A third approach, and I'm really not gonna go through this because it's super complicated, but it's called signups, and this is from Marta Biagali from University of Trento, and she was from Italy. And signups are kind of similar. It's kind of the same concept as these other things. And signups as another type of link RNA that they think that they can manipulate to increase CHD, to increase gene expression level. And she's done a lot of work in CHD8, which is another chromotumor modeler. And now she's also done work on CHD2. And so this is kind of a third potential treatment option. And this is being pursued as well. And so it's just really exciting to see all these different potential treatments for CHD2. And there's probably more potential opportunities out there that weren't even discussed at our conference. And so there's a lot of good ideas, and it's our job to just kind of try to empower these researchers with what they need to push things forward. And so that that was kind of the 4 takeaways. But for me personally, I sort of had a 5th takeaway, and that there's just a great group of scientists working on CHD2. You kind of saw that in the video, and it's just really exciting. And it's kind of one of those things that just gives you a boost of of hope for the future, and just know that there's a lot of people out there that are working very hard to try to get a treatment for our kids. Another thing, and I'm going to sort of conclude with this and hand it back to Marnie, is that while we were down in Florida and kind of after Florida, we started developing, we realized we needed to have a scientific plan. Like, what's our long-term vision of the future? And so one of our board members, Steph Prince, and one of our SAB members, Matt Fuller from Ultragenics, they spent a lot of time putting together a scientific plan, and what do we need to do to drive towards a cure? And so I'm going to hand it back over to Marnie, and she's going to kind of just give you a quick summary of what those costs might look like.
Thanks, Brian. We do have some questions coming in, but I'll let those come in and I'll just finish up here. But yeah, I will just reiterate what Brian was saying, just the excitement that was coming out of there, that there's really potential research being done that's got us, put us in a place of what do we want to do first and how do we want to prioritize these. And so with their help, we put it into kind of a hierarchy where we have what do we think is critical, essential, important, which basically means where do we want to spend our money first. And while we do have some, you can see that research is not cheap. And so while some you can spend anywhere from $25,000 up to, hundreds of thousands of dollars for some of these. And so what we tried to do is prioritize, what is most critical right now and where can we put our funds? And so you can see here what Brian went over today were these iPSCs and animal models. And we've been told many times that we need to have an animal model because while there is possible, they're finding ways to manipulate the CHD2 levels, but we have to test them in animals first. We can't go straight to humans, clearly. So we need to find an animal model. It's critical. And those can run, hundreds of thousands of dollars. And also getting the natural history set up. And so I just wanted to show you here, this is kind of how we've broken it down and about how much money we're hoping to start fundraising. And back to what I was saying earlier about our strategic plan and putting committees together, one big committee that we're trying to fill is our fundraising committee. And so we as a community can start, we do our annual Awareness Day campaign, but we've got to start doing more. So we as a committee and as an organization are going to start trying to come up with other ways, break it down in quarterly fundraising events, things that you can do at home. How can we help you help us? But it's going to take us to raise the money. I mean, we're looking for some of this. We can also try to partner up with pharmaceutical companies to help raise money but our goal as an organization, as a PAG, patient advocacy group, is to get this initial research started. And then hopefully, as it shows promise, then you can get the pharmaceutical companies involved and they can start partnering with us and help fund some of these. But I just, want to make sure that everyone knows it's us as a community that has to raise the money. So these are our goals and we want to share them with you. And I welcome any questions, whether it be organizationally, whether it be scientifically, please, if you don't have questions and you think of them later, you can go through our website to answer any questions. But I will say from a personal level, being in a room with a bunch of scientists that understood what my daughter was going through and could explain it to me. It was just something I could never dreamed of when she was diagnosed 10 years ago. I just feel like it's just we're in a unique time where the science is moving and research is moving at a rate where there's a lot of hope. So hopefully you guys can feel that hope and excitement. And we together as a whole community can help these researchers do whatever they can do to help us. So there you can see those are the ways to donate. And I'm going to try and do some fundraising myself and share with you how it went. And I hope any of you, if you do a fundraiser locally, you share it with us, and we'd like to start celebrating our community members and what you're doing on your own. So please share with us, and so we can celebrate you and we can hopefully build it together. So with that, I would like to go over and look at a few questions that are coming through the chat. And Brian, I'll read some off here and make sure I don't miss any. Laura, I may need your help. But the first question was, has anybody looked at maybe an overproduction of CHD2 instead of an underproduction? And what about multiple genes being affected by CHD2 dysregulation? Did that make sense, Brian?
Sure. So let's see. Yes, as far as CHD2 overproduction, there's research being done right now on sort of studying. There's 3 patients known with CHD2 overproduction. There's going to be a paper coming out in hopefully within about 6 months, sort of describing kind of the characteristics of that. As far as looking at what genes might be dysregulated by CHD2, the one thing, sort of going back to the assembly line, what there appears to be is a lot of little changes. And so those little changes sort of add up to a lot. There was a gene discussed at the conference that in certain cell types, they may have a few targets in mind that going back to Nail Khosry's work of a potential gene that they saw that seemed to be more dysregulated than most. The name of it slips my mind, but some of the work that Nail's hoping to do is to look at CHD2 and a bunch of different cell types and kind of try to figure that out.
Emily, I see you have your hand up, but I'm going to ask this one question that came in the chat first. And it said, is there a way to know which one our kid has underproducing or overproducing by the type of mutation?
So I would say that you could tell that to have the overproducing mutation, there's only, like I said, there's only 3 kids that have that. You have to have that, can only be diagnosed through whole genome sequencing. It's very, I would say that everybody on this call would have the underproducing mutation, but there is a different type of mutation, and Emily actually might be a better person to chime in. But with some CHD2 mutations, it's called a missense variant. And so with the missense variant, you have the right amount of CHD2, but there's one of the copies has a mutation in the protein, and so it doesn't, it might not work correctly. And I think the statistics are roughly about 75 to 80% of CHD2 mutations are these, they would call them premature truncations, where there's a reduction of protein, and the rest are missense variants.
Emily, your hand went away. I don't know if you still had a comment or a question.
Yeah, I was just going to comment on Delia's question. So it is an active question that we're thinking about, the regulation of other genes. It's ongoing, so we're still collecting samples, but that is part of our methylation and RNA sequence kind of omics project.
Emily,
So
would you
hopefully
say
we'll have...
who you are?
Oh, sorry. I'm Emily. I'm a genetic counselor and research coordinator at St. Jude as part of Heather Mefford's group. Thanks to everyone who's participated. I really appreciate it. And thanks for letting me kind of peek in on this call.
Thank you, Emily, for being here. Did you have anything else you wanted to say about that? Okay, because
no, I think that's it. And I think Brian did a good job describing the different types of mutations that everyone with a diagnosis of CHD2 we would expect to have the under expression or what we call haploinsufficiency, but the missense might be slightly different, but I think still under expression.
Okay.
Still learning.
And we may
Add to that, some people might have what is called a variant of unknown significance. This has sometimes come up in the Facebook Messenger group that you may have an inherited case of CHD2, and there's actually a methylation test that you can do to tell if your CHD2 variant is disease causing or not. And it's called the EpiSign test. And if you have a VUS, I highly suggest you talk to your genetic counselor and see if that type of test is appropriate. Sometimes the mutation, they can really tell, like, no, even this is they can tell you, no, you don't need the test. So it's really good if you're uncertain to talk to your genetic counselor, and you can always, also kind of reach out to the folks at St. Jude if you can't get anywhere with that.
Cool, thanks. Got two more questions from Helen Hernandez. She asks, what about small molecules? Has there been a lot of thought given to repurposing? Does the, albeit more complicated, yeast display a phenotype? I'm not as familiar with the yeast.
So the short answer is no. There haven't been a lot of we haven't done a lot of drug repurposing work. That is one of the missions of Neil Khosry's work. The one thing with when you do that type of work on stem cell models, you can't test a bunch of compounds because there's just not enough stem cell material. If we had a good call it zebrafish type model, that work could be done as well. And then she asked about the yeast. The yeast, we haven't. I'm not really aware. The yeast was just more of an example of a gene that was sort of regulated by a long non-coding RNA. They really weren't an apples to apples comparison. So hopefully, not certain how relevant yeast are to, there are other diseases using yeast models to screen drugs, whether that's relevant to CHD2. Not certain about that.
Okay. But she has a follow-up question to that is, what is the MEA equivalent for microglia? What do we look for to understand differences and possible restoration of normal function?
That's a good question. I don't know the MEA equivalent for microglia. Sort of when Nail spoke at the conference, he's like, I could talk for an hour on just certain slides. And I think a lot of what he's doing is he's kind of trying to I know one thing he's doing is he's trying to use various epileptic drugs against these cells to sort of give him clues on what might work. Because he kind of feels that that model is more robust, and it's kind of like not like a wild goose chase.
Okay.
Brian,
Real
would
quickly-
you stop the share for a moment just so people can see each other?
Sure.
Thank you.
This is one that you can answer that's easy to explain. What's a Goldilocks gene? I have a couple people may want to understand what that is.
It just means that if you have too little of CHD2, it causes disease, and if you have too much CHD2, it also causes disease, and possibly and what seems to be a worse disease than having too little CHD2. And so Goldilocks, if you think back to Goldilocks and the Three Bears, you want the amount to be just right. And we think there's going to be a very interesting paper coming out soon about how CHD2 seems to be in species like actually trending down over time. So sort of comparing Neanderthals and human CHD2 seems pretty cool. Steph?
Stephanie.
Yeah.
Wait, I can't hear you.
Steph, you're on mute.
Sorry. I think that was really excellent, guys. And I just want to say thank you very much. And also, I'm sorry I've got a cold, but to go back to the drug repurposing question, we have spoken to a couple of companies about drug repurposing. I think what we have to think about, because we don't have enough money to do this research, so we have to have a strategy, what is going to be most likely to be successful to channel funds when they come into. And overall, when we've spoken to researchers, the general impression is because CHD2 is quite an important gene, drug repurposing is unlikely to make much difference. However, we still did our due diligence and asked a couple of companies, one who does stem cell lines and one who does zebrafish lines. And, you know, just to test 10 compounds is in the order of nearly $200,000. So it's a huge amount of money for what may not give us good results. So we've got that as part of our you know, something we've explored, but it's not like our priority. And I think also when we're thinking about therapeutics, it all comes back down to these outcome measures. What are we trying to achieve for our children? You know, are we trying to reverse their intellectual disability? Are we trying to reverse their seizures, the behavior? And, you know, all our kids are on a spectrum. We all have different challenges. And I think that everybody answering what their particular needs was really helpful. But we've got to see what a therapeutic may achieve. And by going after some of these more sort of, you know, gene-targeted therapies, we may achieve more than going for small molecules. That's what we're hoping for. But excellent questions are coming in.
Thanks, Stephanie. Got some more coming in. Does anyone know, oh, Miriam Fresson asked, does anyone know about research targeting how to get through the blood-brain barrier? Would it be useful to bring people who might research this with our researchers?
That's a good question. I don't know who is working on it, but I know that a lot of a lot of people are working on this. I think it is mostly happening. A huge area is in, when you call gene therapy, and there's people developing, there's various types of genetic editing techniques. One of them is called like base prime editing. And so they're trying to figure out how to get these more permanent therapies to the brain. And so I think that they're kind of the big catalyst for that type of research. I think this is happening all over the world because it's a big problem for everybody to work on. I'm not sure how much of a direct benefit we would get by connecting with those researchers, but it's a good idea. Honestly, I don't know enough about it to really say any more than that.
Just going to quickly say, if you don't mind, it's really difficult to get things past the blood-brain barrier. It has to be a small molecule, or you have to somehow disrupt the brain barrier to get things across. And there's lots of work in oncology, which is and they're still struggling to do it. So with these therapeutics that we've been talking about, even things like gene therapy, which is not an option, they are injected into the cerebrospinal fluid, and that gets, because you're injecting it in there, that's what gets past the blood-brain barrier.
I think there's, I don't know, I think I've seen in SYNGAP might be having a therapy that goes through this cerebral spinal fluid, correct? Yeah, I think so it's an option that's out there that we probably haven't come across yet. Okay. Real quickly, back to the Goldilocks. How is it tested? Is it the EpiSign? EpiSign doesn't test for because the Goldilocks, we really don't know where the perfect balance of CHD2 is. The EpiSign helps understand if your CHD2 variant is actually causing your symptoms, be it seizures or whatever it might be. Is that a right way to explain that, Brian?
Well, the EpiSign test really determines if you have CHD2 haploinsufficiency, and basically there's this type of sequencing called methylation sequencing, and it's just basically like it sequences the whole genome, and it's almost like a visual pattern, and they can see it. And even people with different symptoms will have the same signature. And really, the folks at St. Jude have been kind of instrumental about really like studying the signature in depth. So sort of the Goldilocks is really tested in cells. It's in a lot of diseases, there's something called kind of RNA sequencing. That's sort of an easier way to test whether you have too much or too little of something. But with CHD2, it's really proven to be unreliable, and so they have to test it in cells and really actually measure the amount of protein. And it's a pretty intricate process, and so it's more done at a research level.
Yeah, and just adding to that, but Goldilocks genes, there's a few of them. It's just basically saying where too much or too little causes disease. And we know that the majority of people on this call, whatever variant they have, there's not enough CHD2. What we're trying to do with lots of these therapies is to increase CHD2 up by various means, but we could overshoot and cause disease by overshooting it. And that's why part of the research we need to do is to work out what the range is to make sure it's safe.
The one other thing I would just add, someone asked about genes dysregulated, and we just we solicited patients last year to participate in a research study and to give some blood. And I think we had maybe 15 or 16 patients participate. And so they're looking at, I think it's like 9,000 different proteins that they can measure in the blood. It's called proteinomics. And so we just got the data or in the process of getting the data back on it, and are looking for something that one of these 9,000 proteins is highly dysregulated in the blood with CHD2, and it might serve as like a biomarker to better, like, instead of measuring CHD2, we might look at something else as kind of like a proxy and see, but we have to analyze the data and see if they find anything.
Thanks, Brian. Someone asked, and I want to use this as a reminder plug, that wants to know when the family conference is, and I want to use this. We haven't signed on the dotted line. We are trying to get the costs down as low as possible, and I will also want to throw some thanks out to Justin Schuler, who was on the call. I'm not sure if there he is. He popped up. Thank you, Justin. He's been our lead in the family conference, and we're almost there, but the goal is June 2025 in Denver. So please put those on your calendar. We're hoping the weekend of the 12th, 13th, 14th. It's put that in pencil. But we do hope to be similar. We're going to have St. Jude's there, genetic counselors, a lot of fun. So more details to come. I don't know if you want to add anything, Justin, to that, but I was hoping we'd have information for you today, but we just don't have it yet. So, but yeah, I just want to say thank you to Justin.
Yeah, just trying to push to the finish line, but we're looking to build on the success of our first conference and hoping to have a lot of new faces there and reconnect with people that were there in 23 as well.
Awesome. Thanks, Justin. Tremendous help. And then I was going to say the EpiSign test is in the chat. So if any of y'all want to look into that, there's a link in the chat. And then another question, any knowledge if CHD2 will or is a mention as a part of the various epilepsies with the Epilepsy Foundation in the push for legislators to put more monies in research for epilepsy? And Laura, I believe we signed that.
We did. We signed on to that letter. Whether CHD2 will be, individually looked at, I don't know, but we're supporting for the epilepsy caucuses in the House and Senate. So we're signing on to all the advocacy letters that pertain to anything even remotely related to CHD2 and advancing cures, getting more funding, all that stuff.
Yeah, and I don't remember the exact amounts, but I know the NIH funding is dwindling, and the amount of money out there is not like it used to be. So we are pushing for more funding from the government. Again, back to our plug for our need to do it ourselves as a foundation. So it looks like I don't think I missed any questions, but again, I know you may have some. Our other goal is to have our webinar series bring in some of these scientists to talk more directly and in depth about some of these studies that they're doing. And those will start coming up. They'll post those on the website as our webinars. Please come. We also hope to have webinars just on more what the community needs. So those are coming. So we're really boosting our availability of information that we can provide. So we welcomed your feedback. We welcome any information that you guys need. Please email us. We're here for you. That's the whole reason why we even exist. So anything else you'd like to add, Brian, Stephanie, Laura, or anyone have any questions before we wrap up? Okay, excellent job, Brian. That was great. Thank you so much. And thank you everyone for being here. It's really great to see your faces and your names. And I look forward to our next one. And information coming out soon about the family conference. I'm really excited. So thanks again, everyone, for being here. It's great to see you.
Thank you.
Bye, everybody.
Take care.